Okay, thank you. My name is Binyam Margassi. I'm a professor in the Department of MCDB and OBGYN and Reproductive Sciences here at Yale. My lab studies how chromosomal integrity is maintained in oocytes, which are the progenitors of fertilizable eggs, and how this process fails with age. Okay, so in addition to our shared reason in interest in women's health, one of the major reasons we're here today is actually because of this process, chromosome segregation, which is something that occurs at the start of every human life. In oocytes, chromosomes are segregated with accurate, high accuracy or remarkable precision, but with advancing age, the system breaks down, chromosomes missegregate and cause aneuploidy, which leads to infertility and genetic disorders. We are uniquely positioned to develop interventions for reproductive aging. My lab is a world leader in oocyte biology, having discovered novel mechanisms of chromosome segregation and pioneered a synthetic oocyte aging system. In addition, we also have long-running collaborations with the Yale IVF clinic, which provides direct access to human donor oocytes, as well as pipelines for clinical translation. So maternal aging or reproductive aging is one of the most urgent problems in reproductive medicine. As you'll see on the left here, I can't really point to it, I think this here. So with advancing maternal age, there's an almost exponential rise in the rate of aneuploidy in eggs. And despite massive advances in IVF technologies, we still do not understand or cannot overcome the underlying causes of oocyte aging. And the current approaches largely select around the problem instead of correcting it. So we select embryos, optimize IVF workflows, but still cannot intervene with oocyte aging. And this all happens in the backdrop of increasing rates of infertility globally and also with increasing maternal age. And so we're fast approaching a demographic and medical inflection point. For decades, understanding reproductive aging has been extremely difficult because aging happens or unfolds slowly, and there are several overlapping cellular defects. And so isolating a cause and systematically intervening in it has been really difficult. What our system does is it provides timescale and control. So instead of waiting for decades for natural aging, we can now compress decades of aging in 2 minutes to reproduce cellular defects that are hallmarks of oocyte aneuploidy and aging. So the underlying core technological advance for this actually comes from our work, which mimics in young oocytes age-related cohesion or chromosome cohesion decline. And using in here, you can see in young oocytes before and after treatment with a PROTAC that disrupts chromosome cohesion, chromosomes, instead of being nicely held together, start to prematurely separate and scatter, reproducing a hallmark defect that is seen in oocyte aneuploidy. On the right, we have the underlying mechanism. We use a PROTAC that rapidly degrades a protein called REC8 that holds chromosomes together during meiosis. And this is how we translate the synthetic oocyte aging system into therapeutic discoveries. We will first induce in young oocytes aneuploidies using our synthetic oocyte aging system, and we will apply a focused library of FDA-approved compounds and then measure the rescue of chromosome segregation efficiency using our advanced live imaging technologies to identify compounds that improve chromosome segregation outcomes. And because we'll be screening compounds that are already approved by the FDA, successful hits from this screening will provide a rapid path towards translational studies. Importantly, these are very transient interventions that do not involve any genomic editing or heritable DNA changes. And within the next 6 months, we plan to, or we hope to discover or provide a prioritized list of compounds that can be taken down to translational studies. Current companies in the fertility space, such as Gamito and Cellmatics, largely focused on providing technologies for ovarian support and predictive approaches. And IVF itself largely relies on embryo selection, but these approaches don't really directly address the age-compromised egg itself. Our platform does what others cannot do. It allows us to induce aneuploidy in oocytes and take systematic interventions and measure the rescue of chromosome segregation in real time. And in a global market, IVF market, that is $25 billion, with no approved therapies that directly improve egg quality, this will move our field from describing failure towards fixing it. And over the next 6 months, we plan to test over 3,000 compounds and identify compounds that can improve the outcomes of chromosome segregation that can then be repurposed for improving IVF treatments after completion of these clinical studies. And this is how we plan to use the budget. And our goal is simple. Instead of observing reproductive aging, we plan to functionally intervene in it. And so that's the overall aim of this project. I'm very happy to take questions.
Thank you. Judges?
Thank you very much for that. Can you share a little bit more about what you think the business model for this is and how you could potentially either partner with large pharmaceutical companies or even, I know there are some smaller companies that are looking at this problem that have kind of, specific modalities that are addressing equality. So just would love to understand kind of how you're thinking about that.
Yeah, so the immediate application of this would be in an IVF setting, basically improving the quality of eggs that are being used for IVF treatments. And the clinical precedents are clear. There are products out there currently that help to improve the reproductive outcomes, such as antioxidants, but these don't directly address the root causes of aneuploidy or chromosomal abnormalities. And this is where these compounds would basically be applicable. So we would be looking at improving kind of like basically having an IVF supplement that improves the quality of eggs for better outcomes of IVF clinic treatments.
And the timeline here to identify these candidates is about 6 months. And
yes.
Do you find that or think that you could reduce that timing, or is that sort of just how long it'll take?
Yeah, so I think currently we already have the system set up. So the mouse model is basically what we innovated in my lab. So we don't actually have to generate it. So we only need to basically purchase those compounds and do the screening. And we have a way for high throughput imaging. That's also something that my lab specializes in. So we'd be able to proceed within the next 6 months to identify candidates.
Building on how this therapy would be delivered. So are you envisioning it's delivered to a female?
In the short term, it would be an IVF supplement. Basically, once the eggs have been isolated from the patients and they're being prepared for fertilization in vitro, they would be treated with these compounds. But
so
in
it's
the long
the eggs
term,
that would be treated. It's not the woman that would be treated.
Yeah, and that's the thing, because the regulatory pathways, as we were discussing early, are even crazier when it comes to actually editing the genome of an egg, and this does not involve any of that. So we would be dealing with the L site before it is implanted into the patient.
Okay, and then to that effect, is there an easier way? I mean, it's not trivial to develop a PROTAC molecule the way that you've got it outlined there. Is there another?
So we don't actually have to develop the PROTAC. So the PROTAC, we already have it to create this aging-like phenotype that we can then try to decelerate using these FDA-approved compounds. So because then we would be, whatever we identify, we would be repurposing and doing the chemistry on to improve them, but we don't actually have to come up with a new compound.
So you are planning for new composition of matter. It's not that you're trying to repurpose something that's already out there.
Yeah, so I mean, these are approved compounds, so if we do need to repurpose, we do have the structures, and then we have the chemistry that we can use to improve them.
Okay.
Thank you.